Welcome to Cancer Lesson 3.3, How Can the Immune System Cause Cancer? The goals of this lesson are to explore how inflammation regulates cell proliferation, consider how chronic inflammation can lead to DNA damage, and investigate how chronic inflammation during obesity may lead to cancer. At the end of the lesson, the students should be able to explain the difference between normal inflammation and chronic inflammation. Describe the role of secondary factors in converting normal inflammation into chronic inflammation, and explain how chronic inflammation can lead to cell proliferation and DNA damage. We will achieve these goals by engaging in an interrupted case study exploring the link between chronic inflammation, obesity, and cancer. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. The immune system responds to both tissue injury and infection with inflammation, which stimulates cell proliferation. When inflammation becomes chronic, the increased levels of reactive oxygen species produced can lead to DNA damage. Several secondary factors that turn normal inflammation into chronic inflammation are associated with cancer. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure. The student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet. The key points of the do now are that inflammation causes cytokine secretion that leads to cell proliferation in order to heal the tissue damage. Normally, inflammation stops when the tissue is healed. Chronic inflammation acts as a carcinogen because it produces reactive oxygen species that damages the DNA of the proliferating cells. The do now harks back to lesson 2.3, which used wound healing as an example of the types of signals cells produce when they need to start and stop dividing in order to heal wounded tissue. First, the environment of the wound starts to recruit immune cells, and these immune cells secrete growth factors that stimulate cells to proliferate. The immune response to this wound is called inflammation, but cells don't need to proliferate forever. When the tissue is repaired, proliferation should stop, so there must be stop signals. Students should remember that cell contact is a major way that cells know when to stop proliferating. In healthy tissue, once a wound heals, the inflammatory response switches off. But what happens when the inflammatory response does not stop? Not only does the wound not heal, the chronic inflammation can act like a carcinogen. How does this happen? Recall that carcinogens cause mutations in genes that control the cell cycle and death. These mutations can result in cancer. But how does chronic inflammation act like a carcinogen? Inflammation produces reactive oxygen species in order to kill pathogens. These reactive oxygen species damage DNA. If cells with damaged DNA proliferate, they can be transformed into cancer cells. Inflammation becomes chronic when growth factors continuously stimulate cells to proliferate. At the same time, the reactive oxygen species are produced that can damage DNA. Hence, mutated cells may also be stimulated to proliferate. But what makes inflammation chronic? The growth factors damaged cells release to stimulate the inflammatory response are called cytokines. One of these cytokines is tumor necrosis factor. Normally, when the wound heals, cells stop secreting TNF, and therefore inflammation subsides. But secondary factors can also stimulate secretion of TNF, so inflammation becomes chronic. Examples of secondary factors that can stimulate chronic inflammation are listed on this slide. In the activity, students will work in groups to complete an interrupted case study on the relationship between obesity, chronic inflammation, and breast cancer. In the case study, students will read the results of experiments conducted to investigate the role of obesity in breast cancer. They should pay attention to the study design. This case study will also incorporate concepts learned in previous lessons. We collectively interpret the results of the experiments in the wrap-up. Students should have deduced the following sequence of events. Enlarged fat cells found in obese tissue release more TNF than normal fat cells. This TNF causes inflammation in the obese tissue and proliferation of the epithelial cells in response, even though they are not strictly damaged. They in turn release more TNF. This is called a positive feedback loop and results in chronic inflammation. The chronic inflammation will cause DNA damage in the epithelial cells. If the mutations are in important genes that regulate growth, this could lead to cancer. The discussion concludes with a list of cancers that are linked to inflammation and the secondary factors that can induce chronic inflammation. Note that alcohol is an important secondary factor in a number of different tissues. For homework, have students read an article about how aspirin may protect against cancer. Students often have difficulty understanding why chronic inflammation creates the perfect carcinogenic storm. First, chronic inflammation produces both pro-proliferation signals and also carcinogenic free radicals. The cells in this environment are therefore constantly proliferating while being exposed to carcinogens that promote mutations. This leads to transformation. Another concept students struggle with is the positive feedback loop of TNF-alpha in the worksheet. The point is that the cell is able to make its own TNF-alpha. Initially, inflammation switches on TNF-alpha production, but once inflammation stops, the cell's own loop continues. The effect of this is that the cell resists apoptosis. At the end of the lesson, collect the lesson worksheets to assess students' understanding of the TNF signaling pathway from stroma to parenchyma and their understanding of why one would look at RNA versus DNA to compare protein levels in tissues. Therapies that curtail the overactive immune system are becoming very important in cancer. We will look at them in more detail in the next unit. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.